Hello. Today we start our course Biology 281, Human Form and Function, or in other words, it's human anatomy, so what the body is composed of, and physiology, how the body works. So we'll talk about the functioning of living beings. But before diving into the details, how do we even know that humans are living organisms? And you can stop and think about it because the answer is not easy. Or if you don't want to do this, I can tell you. So first of all, it's ordered complexity. So what does it mean? It means that, yes, we're made of molecules, but we are not just a heap of molecules somewhere on the floor. Our molecules are organized in a very particular structures that forms the cells. And then cells, they combine together into various tissues, and tissues form the organs, and organs form the organ systems in which each particular organ follows the other, and they work together to complete their function, like digestion or reproduction. So it's all very organized. Humans, like all other living organisms, are capable of reproduction. So creating the offspring. Growth and development, that's another function of or characteristics of living organisms. And to provide this growth and development, we need energy. So we are able to process energy, to obtain it, transform, and to use it in the body. We are also sensitive to the environment and we respond to it. And as an example, you touch something hot and you withdraw your hand immediately. So you got the signal, you sensed it, and you responded to it. Our environment changes all the time, but we are also capable to maintain our internal conditions. So for example, it can be very hot outside in summer, can be very cold in winter, but our body temperature stays the same. 37 degrees Celsius. We are organized out of cells, and we'll talk about this later, but cell is the unit of life. So to qualify as a living organism, you have to be made of cells. We are also subjects, together with all living organisms, we are the subjects of evolution. So we adapt through many generations, but we adapt to our environment. So we'll study the humans, but how do we do this? And there are many ways and many levels at which we can study living organisms, starting from a very low level of atoms and going through the molecules and cells and organs and populations, or we can study humans in the context of the entire biosphere, like as a part of everything that happens on the planet Earth. And each level is more complicated than the other, and each level has its own properties. And sometimes it's impossible to deduce the properties just by looking at the previous levels. Okay, maybe I know how the cells work, but that wouldn't explain me the distribution of or propagation of disease in a population. So we have to study each level separately, but also in connection to all other levels of human organization. But for this course, we will mostly concentrate on the cells. I'm sorry, I'll do the pointer. Cells, tissues, organs, and organ system, and organisms. Mostly because that is mostly related to the topics of human anatomy and physiology. But still, we'll start with the lowest levels, with atoms and biological molecules. And you might ask why, because I just told you that you cannot figure out how the organism works by looking at the atoms. Maybe not, but you still can figure out a lot of it. And that was the argument of many scientists in the previous centuries, like, for example, in 18th, 19th century, when the physics and chemistry they were very popular sciences. And many scientists believe if you understand the physics behind the processes that happen in human body, or in living organisms, or chemistry behind it, you will eventually explain the nature of life. And they argued with the other group of scientists who believe that it's not true because the living organisms are completely different from non-living objects, because they contain some element, some vital, they called it the vital spark. And that led to the development of the theory of vitalism, the vital spark theory. And one of the ideas of this theory was that cells have inside something, some substance they called the protoplasm, which was responsible for all chemical reactions in the cell. And if you break the cell, you will disturb the protoplasm, and you will eliminate or you kill the vital force. Why that was not a good idea? Because that essentially canceled any studies made outside of cells. So biochemistry, the science, would be impossible, or molecular biology, because if it's not in the cell, you cannot study it. It's like doing this would be senseless. But this idea was successfully confronted by the German scientist Eduard Büchner. And what he did, he studied yeast, and yeast are known to ferment sugars. So if you take yeast and mix them with cane sugar or with glucose, the yeast start to ferment it through chemical reactions. So they will convert sugar into alcohol and carbon dioxide. If you mix it with sugars that yeast cannot process, like lactose or mannose, nothing will happen. So he suggested that chemical reactions in living organisms do not really require living cells. They can be performed by the substances inside the living cells. So like no vital spark. So what he did, he took yeast and broke them, just ground them with quartz and diatomous earth to break the cells. And to be sure that all the cells are broken, he put them in the press 400 to 500 atmospheres pressure, incredible pressure, definitely destroyed all the cells and squeezed the content outside. So he took this content and no living cells in it, just the content of the cells themselves, and mixed it with sugars, and he got completely the same reaction. Cane sugar and glucose were fermented, and lactose and mannose were not. So he proved that you don't really need the live cells. You can study a lot of processes that happen in live cells, but by like outside. So in the test tube. And that's why we actually do study cells. We study a lot of processes in biochemistry, just the reactions that happen in cells and molecules and atoms involved in these reactions to understand how life processes work. Just one thing I wanted to tell you, if you notice, I have two types of slides. I have slides on the white background, and that carry the information that I want you to learn. And there are a lot of slides that will be on this, beige, yellowish background. And these information, you definitely have to look at it, you have to listen to it, but you don't have to memorize it. This is more to give you examples or to extend the material to make it you understand it better, but you don't have to learn it. So what do we have? What is our body consist of? So there are 118 elements in the periodic table, 91 you can find in nature, but only 12 of them are represented in the body in substantial amounts. And here is the list of elements that you can find in decent amounts in the human body. There are also a lot of elements that are present in the so-called trace amounts. They are very important, like for example, iron for carrying the oxygen, copper, it's a part of some enzymes, or iodine is a part of some hormones, but their content is very little. And out of these 12 elements, 96.3% of weight belongs to carbon, hydrogen, oxygen, and nitrogen. It's kind of obvious why hydrogen and oxygen, there's so much of these two elements, because H2O is water, and our body is like about 70% consists of water. But why carbon? And this is because the molecules we are built of are biological molecules, organic molecules, and organic molecules are carbon-based. So what does it mean? If you look at this molecule, you see C, 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 C. Here, C stands for carbon. The backbone of organic molecules and biological molecules is carbon. And it's kind of a common knowledge in organic chemistry and biochemistry, to the point that scientists do not even write these C carbon atoms in the molecules. Simply, assume that the carbons are here. So if you see most molecules, you will not actually see letters C in them, because it's implied there's a carbon here. What I showed you here are pretty simple molecules, small. Cells use them to build even a larger number of other molecules, of big molecules. And these big molecules are called macromolecules. Macro for big, because of their size. They are also called polymers because how they are made. So they are made of monomers. Mono stands for one, so units, single units, and they are kind of organized together, linked together in a bigger substances, bigger molecules. They can be done from identical blocks, they can be done from different blocks, but the idea is you take something simple and then you repeat it many times in a particular sequence, and you have big polymeric molecules. And you see how many opportunities this method implies, because, when it's the same molecule, you can have different lengths. When it's different molecules, it's a little bit like beads on the string. You have a limited number of beads, but the combinations can be unlimited. It's the way you string them. So there are thousands, maybe hundreds of thousands of different molecules in the cell, but they all can be classified into 4 major types. It is carbohydrates. And here there's some, like the produce that has this type of molecules in large amounts. Proteins, lipids. Lipid is a scientific name for fat, and nucleic acids. And we'll start with carbohydrates. Carbohydrates, sometimes they're called sugars, are molecules that contain carbon, hydrogen, and oxygen at a very particular ratio, 1 to 1, 1 to 1. 1 carbon, 2 hydrogen, 1 oxygen. The name carbohydrate comes from this structure. It's carbon and water. Carbohydrate. So, kind of. Sometimes you can guess that the compound is carbohydrate. It ends with O's. Very typical. Glucose, sucrose, fructose, lactose, cellulose. Not always, but very often. So what are the functions of carbohydrates? First of all, they are a source of energy. We burn glucose to get energy for all our cells. They can be used by building blocks of larger molecules. Like simpler, smaller carbs are used to build bigger carbs. They are used as storage molecules, like starch in plants and glycogen in humans. And they can be structural components of the cells, like a typical example is cellulose in plants, which cellulose is a big component of the cell wall, what makes them kind of rigid, the plants. So how can we classify carbohydrates? We can do it according to size. And there's simple sugars, or the scientific name is monosaccharides, which really means one sugar. And they consist of just one sugar unit. Oligosaccharides. Oligo means kind of several. Can be di, tri, tetra, penta, 2, 3, 4, 5, up to 12 units. And very typical biological molecules are polysaccharides, when you have more than 12 sugar units. Again, poly means many. I'll start with monosaccharides. You see they're simple, just one unit, one. You definitely heard about glucose, fructose. There are also others. They're used as source of energy. I was talking about glucose. And also as building blocks for other molecules. For example, these two are parts of nucleic acids. In humans, glucose is the main source of energy. Where can you find monosaccharides? Like, for example, honey. Honey is a mixture of fructose and glucose. Or if you want pure fructose, it's corn syrup, high fructose corn syrup. Many people see it as something really harmful. I wouldn't say this. It's not the syrup itself. It's more like that is put in every single, candy produce. It's just too sweet. We consume it too much. It's not the problem with syrup itself or with fructose itself. It's mostly because it's overdone. Disaccharides. Di stands for two. So it's formed by two monosaccharides linked together. And I'm just giving you two examples because you are very well familiar with them. Maybe you didn't know that they were disaccharides, but they are sucrose, which is our regular table sugar, just glucose and fructose linked together. And another sugar is lactose, or milk sugar, also because many people are intolerant to lactose. They cannot digest these disaccharides. Polysaccharides, monosaccharides linked in a long chain. They are used for storage. In plants, this is starch. In animals, and humans, it's glycogen. I'm just want you to remember the name because I will be talking about it much later. And just a structural element like cellulose in plants. So the structure of polysaccharides and the properties depends what units they have in them and how these units are bonded together. And as an example, I want you to show 3 polysaccharides. They're all present in this picture. And one of them is starch, glycogen, and cellulose. And they are all made of glucose. All these units are glucose. But when it's just kind of linear, it's starch. When it's branched, it's already different compound, it's glycogen. And when the same unit but linked a little bit differently, and it forms cellulose. So very smart way of nature when it uses the same building block, the same molecule of glucose, and just manipulating the structure or the links, it creates 3 completely different polysaccharides. And they're all present in the picture because we have starch in the potatoes, we have glycogen in the mussels, and we have cellulose in plants. So I will continue in the next part.